a new topic in physical anthropology in paper 1. So it is unit 9.1 human genetics. Unit 9.1 in paper 1 anthropology. So this comes under physical anthropology. It is unit 9.1 human genetics. In this chapter earlier we have discussed pedigree analysis, isn't it? That was the topic which we discussed few days back. Under pedigree analysis we discussed X dominant, X recessive, autosomal dominant and all this, isn't it? So we have finished that topic. Apart from that, we have other topics left, that is the foster child twin study, co-twin method, cytogenetic method, chromosomal analysis, karyotype analysis, biochemical method, immunological method, DNA technology and recombinant technologies. So in the end of this chapter, you can find these terms DNA technology and recombinant technologies. Actually speaking, there's nothing different called as DNA technology and recombinant technologies. It is a single term, recombinant DNA technology. Okay, so please do not get confused that there is something called as DNA technology which is different from recombinant technology. See, our people who set up the syllabus, our examiners are so lazy that they could not even rectify this mistake. So somewhere from some textbook they would have picked out this syllabus and they would have made it. So exactly whatever was there in that book, they have just copy pasted it, isn't it? So there's nothing like DNA technology and recombinant technology. They both are not separate, they are separate units. It is a simple term called as recombinant DNA technology. Okay. So in short, we call it as RDT. Okay, in short terms, we can call it as RDT or recombinant DNA technology, and it is also called as genetic engineering. So genetic engineering and recombinant DNA technology both are the same. Fine. So we shall discuss recombinant DNA technology today. What is recombinant DNA technology? Recombinant. So the word or the term recombinant itself means we are recombining two different things together to produce something new, isn't it? So recombinant DNA technology, the technique in recombinant DNA technology is the transfer of a sequence of DNA from one organism to another organism. Hence, it is also called as molecular cloning or gene cloning or genetic engineering. So there are many terms for this recombinant DNA technology. So in the examination, say for example, they do not straight away ask recombinant DNA technology. Instead, they ask a question saying explain genetic engineering or molecular cloning or gene cloning. Please do not get confused. Don't think ma'am has not taught me gene cloning or molecular cloning. That is something different from recombinant DNA technology. It is not. Okay, recombinant DNA technology, gene cloning, molecular cloning, genetic engineering all mean the same. Fine. So when discussing epidemical anthropology and other topics also, we spoke a lot about diabetes mellitus, isn't it? Diabetes mellitus, which is a common disease which is found among the elderly population worldwide. So it is not some disease which is specific to a region, it is commonly found worldwide. In common terms we call it as sugar, isn't it? Otherwise scientifically it's called as diabetes mellitus. So what happens in diabetes mellitus is the pancreas is not capable of producing sufficient amounts of the insulin hormone which is necessary for breakdown of the glucose in our blood. So whatever food which we consume, that is the carbohydrates which we consume, ultimately gets broken down to glucose, and that glucose needs to be broken down. The function of breaking down glucose is carried out by an enzyme which is secreted by the pancreas. This enzyme is called as insulin. So what happens is after a certain age of 40 years or 45 years or 50 years, the pancreas failed to produce sufficient amounts of insulin which is necessary for the breakdown of glucose, as a result of which there is increase in the sugar component. There is increase in the sugar levels in the blood, and that leads to the disease called as diabetes mellitus. So here what usually doctors will prescribe is, in the initial stages when we have mild diabetes, doctors will prescribe tablets which goes and which goes and affects the pancreatic cells and makes it secrete the insulin. So this is how the tablets function. So it goes and targets the pancreatic cells so that it will produce insulin. But later, when the insulin level goes high, suppose you cross around after 200, we know how insulin is measured, right? So we have tests done before food. Okay, early morning you can find most of the diabetic patients, they'll go around 7 o'clock or 6 o'clock and give their blood once for testing. Then they will have breakfast. After 2 hours after breakfast, once again they will go give the blood for testing. So 2 times they will measure and they will tell what is the sugar level that is present in the blood. So suppose when it crosses about 200 or about 300, for in some cases it goes up to beyond 300 also. In those cases, usually the doctors will prescribe insulin injections. So how do you think this insulin is manufactured? How is it synthesized? So what the insulin, when our pancreas, our own pancreatic cells are not capable of producing sufficient quantities of insulin which is necessary for the breakdown of glucose, we have to rely on external sources. That is why doctors will prescribe us to take injection, insulin injection. So it is nothing but the insulin hormone which we inject into our body, and that breaks down the glucose in our blood. Okay, so how do you think, have you ever imagined how this insulin was synthesized for the very first time? Well, how did they actually make it? So diabetes is a disease which has been present since several, you can even call, we don't know how many years it has been present. Even more than 100 years it would have been present, even I am not sure. So initially, older days, what they used to do was, whenever a person wanted insulin, like how was the insulin manufactured during the olden days? So what they used to do was, they used to extract the insulin from cadavers. You understand what's the meaning of cadavers, right? Or from animal sources like pigs. So what they used to do is, they used to slaughter animals like pigs, and from the pancreas of the pigs they used to extract the insulin, and further they used to process it in industries, in manufacturing industries, that is in what do you call that? There's industries which make medicines, there they used to process it, and further that hormone used to be injected into the diabetes patients. So there were 2 issues which cropped out of this. First and foremost, that is the animal hormone which you are extracting it from the animal body, right? So in many patients what used to happen was, they used to have an allergic reaction because it was not very much similar, though it is similar to the insulin produced in our human body, because it is of animal origin. The people who took this insulin started showing severe allergic reactions. That was one case. And one more thing was, there were a lot of ethical issues involved in this. So we know that in our country, there are, pigs are considered as, pigs are worshipped by certain communities, isn't it? So again, there was a ethical issue also in taking hormones which were extracted out of animals. So these were the primary issues which the people faced. First, they started developing allergic reactions, and secondly, because of the ethical issues, many people were hesitant to go ahead with this insulin which was extracted and which was derived from the animal sources. So now they had to find some other solution. So it is that time they took the help of recombinant DNA technology to produce insulin hormone in the laboratories. Okay, so the very first, how many of you know when the commercial form of recombinant insulin was produced, in which year, and what was the name of that insulin? Can any of you tell me? Okay, before? Okay. Meanwhile, as and how we are discussing this, if any of you know that, you can just let me know. So when was the first insulin, that is recombinant insulin, was produced commercially, and what is the commercial name of that, and what is the company which produced the first recombinant insulin? Just let me know. If not, by the end of the class I will tell you. So how do they produce insulin? By using recombinant DNA technology. So what they did was, they took the gene, they identified the gene which is responsible for producing the protein which is called as insulin in humans. Okay, they extracted that gene. So what they did was, so from human beings, they extracted the gene which is responsible for producing the insulin. Say there is this gene which is responsible or which codes for the protein insulin. So now you might be wondering, just now ma'am said insulin is a hormone, now she's telling it is a protein. See, hormones are also proteins only. Okay, so what they did was they extracted the gene, say this is the gene which is responsible for or which is coding for the hormone insulin. They extracted this gene from the human beings. Then they put this gene into a bacterial cell. Okay, they put this gene into a bacterial cell and they cultured this bacterial cell and they grow lot number of colonies, thousands and lakhs and millions of colonies of this bacterial cell. Okay, so what happens is when this gene is transferred into a bacterial cell and it is multiplied, it is cultured, it is grown in laboratories, then it produces large quantities of insulin because this gene gets expressed inside the bacterial cell. And after you get clones, that is millions and millions of cultures of this bacterial cell, from that bacterial cell they will extract the insulin. They will extract the insulin hormone and they will further process it in the pharmaceutical industries, and that insulin they will administer to the diabetic patients. So this is how they started using recombinant DNA technology for production of insulin hormone. So I just gave you this example. That doesn't mean recombinant DNA technology is all and all concerned only about production of insulin. Recombinant DNA technology itself is a huge topic, is a huge subject. So when we studied in MSc and BSc also, we had one full semester, one topic, one subject was recombinant DNA technology. Okay, so for more than 1 year, 1 year in BSc and more than 1 year in MSc, I have studied several books on recombinant DNA technology or genetic engineering. So there we study much in depth, whereas here from anthropological perspective, as per our anthropology syllabus, I will be explaining it in very simple terms and only very little things which you need to know. You need not go too much into depth. Fine? Okay, so now I will show you how this process is done with the help of pictures. So in this chapter, first we will discuss recombinant DNA technology and then further we will move on to the other topics like twin study, co-twin method, biochemical method, immunological methods, chromosomal and karyotype analysis and so on. Fine. First topic we shall discuss is recombinant DNA technology. Yeah. So recombinant DNA technology or genetic engineering involves three primary steps. First one is gene isolation. So this is the gene which is necessary for us, right? So I will explain this with the example of the insulin hormone so that you will be able to understand it properly, and then further similarly you can understand the other applications of genetic engineering also. So first, the gene which we are interested in. So in this case it is the insulin, the gene which is coding for the hormone insulin. That gene is called as the gene of interest. So this gene is extracted from the host organism, that is we, from human beings. We will extract that gene which codes for insulin. So that is called as the gene of interest. So here in this case, this is the gene of interest. Okay. And then we have a vector molecule. So now if you have to transfer this gene into the bacterial cell, you cannot straight away take the gene and put it into a bacterial cell. You need something, some vehicle which will carry this gene into the bacterial cell. Say for example, you have to reach a college. So you are at home, you have to reach a college and you need something which can carry you to that place, isn't it? Say you take your bike or you take your car or your college bus will come and pick you. So what is that? That is a vehicle which is carrying you from your home to your college, isn't it? Similarly here vector is a vehicle which is going to carry this gene of interest into the bacterial cell. Okay, so what we will do is first we will isolate the gene of interest which we need, and then we will take a vector molecule. Most commonly used vector molecules are plasmids. Okay, plasmids are self-replicating DNA molecules which can actively replicate inside the bacterial cell. So this is again a DNA molecule only. So in bacterial cells you can find some circular DNA molecules which are present. Those circular DNA molecules which are present in the bacteria are called as the plasmid. So these are capable of self-replication, that is on their own they keep on replicating. So that is called as the plasmid. So plasmids are the most commonly used vectors or vehicles. Okay. So you understood what is the meaning of vector, right? Vectors are a vehicle which enable us to transfer this gene of interest into the bacterial cell. So it acts as a vehicle which carries the gene and enables it to enter into the bacterial cell and multiply over there. So this is the vector and this is the gene which we are interested in. So now we have to combine these two. We combine them to form a recombinant DNA molecule. Here you can see this is the vector molecule. And here we are taking this fragment DNA and we are attaching it with it to form this recombinant DNA molecule. This is called as the recombinant DNA molecule. So here we have the plasmid and we have the gene which we are interested in. That is, in this case, the gene is coding for the insulin hormone. So that gene we have inserted over here inside the plasmid or the vector. Then finally, now we have to transfer this recombinant DNA molecule into a bacterial cell. So this is the bacterial cell. We transfer this into the bacterial cell. So here the second step is transportation of the recombinant DNA molecule into the host cell. So the host cell here is bacteria. Okay, so earlier I said the host in the sense while extracting DNA, that is we are taking the DNA for insulin hormone from us. Here the host cell is bacteria. Understood? So here the bacterial cell is there and we are transferring this recombinant DNA molecule inside the bacterium. So what happens is after the recombinant DNA molecule enters into the bacterium, it starts multiplying. It starts producing several copies of itself. Okay, here you can see it is starting producing several copies of itself. So from one recombinant DNA molecule, several copies of the recombinant DNA molecule is produced. Got it? Further, even the bacteria also keeps multiplying, isn't it? So again you can see here, bacteria also keeps multiplying in number, and as and how the bacteria multiplies, even the plasmid within also will keep on multiplying. Got it? So within the bacterial cell, the plasmid also keeps multiplying several times, and even simultaneously the bacterium also keeps dividing again and again, producing several copies or several bacterial colonies are grown. So what happens inside the bacterial cell when this recombinant DNA enters and it starts replicating, it starts expressing itself. That is, it starts expressing itself and it starts producing the insulin hormone over here. So there is large quantities of insulin hormone produced in every single bacterial cell. Further, the bacteria is also multiplying several times, and we have several colonies of the bacteria over here. So finally, if you see, there will be thousands and millions and billions of bacterial cells which are producing that insulin hormone. So now from this bacterial colonies, we can extract that insulin hormone and we can process it further in the pharmaceutical industries, and we can have the final insulin which can be injected into the people who are suffering from diabetes. Okay, so recombinant DNA technology involves three important major steps. First one is isolation of the gene of interest, that is the gene which we want to express. Okay, so this might be some human growth hormone, the gene for some human growth hormone, or it might be the gene to produce some other character. Okay, any gene. So that gene we have to extract or isolate. That gene is called as the gene of interest. Then we have to select a vector. Vector is the vehicle which enables us to carry the gene of interest into the bacterial cell. So mostly you can find that there are cloning vehicles or carrier molecules. Plasmids are the most commonly used vectors. Fine. So plasmids are circular molecules of DNA that lead an independent existence in the bacterial cell. So nowadays we also have technique of injecting the DNA straight away into the bacterial cell. That is also there. But in genetic engineering, most commonly we use vectors. Okay. Then within the host cell, the vector multiplies. That is, this recombinant DNA starts multiplying within the bacterial cell, and along with that, the bacteria also starts dividing and produce several millions and billions of colonies of the bacteria, where that gene which we inserted gets expressed and that particular protein which we are looking for is produced. Then we can extract that protein. So in this case, it is the insulin hormone which is getting produced. We can extract that insulin hormone and we can process it further in pharmaceutical industries to produce the final insulin which we can use it on patients suffering from diabetes mellitus. Okay. So these are the various stages of recombinant DNA technology. Okay, first is isolation of DNA or the isolation of gene of interest, and then taking a vector molecule which is suitable for this, and then combining both of them to form the recombinant DNA molecule, and then this recombinant DNA molecule is inserted into a bacterial cell. This is plus. So recombinant DNA molecule and bacterium are combined together. So the recombinant DNA molecule enters into the bacterial cell, and inside the bacterial cell it starts multiplying, producing multiple copies of itself. And then along with that, even the bacteria also multiplies, and finally we have large number of colonies of bacterium which have produced that hormone or the protein which we were interested in. That can be extracted and it can be utilized further. Got it? Don't worry, I will be sharing all these pictures on WhatsApp. That is why I keep telling you, if anybody has not joined the WhatsApp group, join it. I'll show you another picture also. This is also same picture. Here, this picture. So first, here you can see this is a bacterial cell which has the plasmid. So first the plasmid is isolated, okay, and plasmid is kept ready and it is cut with the enzymes. So now in very common terms, very simple terms, I told you that the gene is isolated from the whatever gene which we need, which is taken from the humans, and then finally the plasmid is taken from the bacterial cell. Both of them are recombined to form the recombinant DNA molecule, and that recombinant DNA molecule is inserted into a bacterial cell, where the recombinant DNA molecule multiplies and produces several copies of itself. In addition to that, what happens? The bacterial cell also divides, producing several copies of the bacterial cell, and there the gene of interest gets expressed, and we can isolate the protein from that and we can process it further for usage. But here, how easily I told you, the gene will be cut, the bacterial plasmid will be cut, and they both will be joined. Is it so easy? So just like, so what there are, we need various enzymes to carry out these functions. First and foremost is the restriction endonucleases. So what are restriction endonucleases? Restriction endonucleases are also called as molecular scissors. Say you want to insert a gene into this plasmid. You cannot just take it like this and do it, right? So first you need to cut this. You need to cut this, then only you can insert the gene over here. So to cut it, like how if we need a paper, okay, if you have to cut a paper, we need a scissors to cut it, right? So if we have a circular paper like this and we want to add something else into this like this, what we will do, we will take a scissor, we will cut it, we will open it up, then we will add another new piece and we will put glue on both sides and we will seal it. Isn't it very simply? I'll show you. See, we use many enzymes to carry out these processes in recombinant DNA technology. The first one is the restriction endonuclease. Say you have the bacterial plasmid and you have the gene of interest. Okay, this is the gene of interest and this is the bacterial plasmid. Now you have to recombine these two. That is, you need to cut open this and you need to include this. Wait, let me put it in other colors so that you can understand. So first, say this is the bacterial plasmid, okay, and this is the gene of interest. Okay, this is the, again it is coming in black only. Fine, wait then, I will do something else. Now say this is the bacterial plasmid, okay, and this is the gene of interest. We have to combine these two. So what we have to do, we have to open this bacterial plasmid like this, okay, we have to cut it open and then only we can put this here, okay, then we have to put this here like this, okay, and then only it will recombine to form a plasmid molecule like this. So this will be the plasmid, this entire, this will be the gene which we have added newly. This is the recombinant DNA molecule, isn't it? So how do we do this? We have to cut the plasmid, we have to cut the gene of interest. That function is carried out by an enzyme called as restriction endonuclease. Okay, just like how we use scissors to cut paper, isn't it? Suppose you have a paper ring like this and you want to add another paper like this. So what you will do, you will cut it open over here, then you will take this, you will paste it over here, on both sides you will put glue and seal it. Then only it becomes a circular proper molecule which has the new paper which you want to add. Similarly here, this plasmid is cut open by restriction endonucleases. These are the enzymes which act as scissors to cut the plasmid. So they do this function. Then again, after adding the gene of interest, we need to seal both the sides. We need to put glue and stick them, right? So the function of the glue is carried out by enzyme called as ligases. So restriction endonucleases are the enzymes which cut the DNA. And a very good example for restriction endonucleases are DNA ligase or DNase 1. This is a very good example of a restriction endonuclease. Then we need enzymes which need to fix the ends of that, isn't it? Just like how we put glue and seal both the ends, similarly here also we need some glue to seal both the ends. So that function is carried out by the enzyme called as ligases. Okay, ligases are the enzymes which seal or join the DNA fragments. So one good example of ligases is T4 DNA ligase. So under restriction endonuclease also we have several restriction endonuclease. So there are specific endonucleases which cut at a specific site, all these things. So that goes, see, so we had separate books on restriction endonuclease itself. Okay, how exactly they identify, so whether they cut at the terminal ends or in the center, what is the sequence which they identify, all that. So we do not go so much, we are not studying genetic engineering here, or we are not studying biotechnology over here. Okay, we are studying this as part of anthropology. So in very simple terms I am explaining it to you. So ligases, very good example of ligases, T4 DNA ligase. Okay, then polymerase enzyme. You all know what is polymerase enzyme. So polymerase enzyme is the enzyme which synthesizes a new strand of DNA complementary to the already existing DNA or RNA template. So what does polymerase enzyme do? So suppose there is a DNA strand over here. It has A, T, C, G, T, A, T, T, C, C. So if you add polymerase enzyme and few nucleotide primers, then it will synthesize a strand complementary to this. So what DNA polymerase will go do? It will add TA, GC, A, TT, AA, GG. So it will synthesize a complementary strand to the already existing strand of DNA. That is the function of polymerase. We already discussed about polymerase in DNA replication, protein synthesis, transcription, translation, isn't it? Fine. So these are the important enzymes which we utilize in recombinant DNA technology. First one is the restriction enzyme, which is also called as molecular scissor, which is utilized to cut the DNA. And then ligase enzyme, which is used to seal the DNA or to join the DNA fragments together. And then we have another enzyme called as polymerase enzyme, which functions to synthesize a new strand of DNA complementary to the already existing template DNA. Got it? Now I'll show you the picture so that you will understand better. So again I am explaining the same recombinant DNA technology method, how it is done. Earlier I showed you another picture and explained. Now I am showing you a different picture. So this is a bacteria which has plasmids. So that bacterial plasmid is isolated or extracted from the bacterium. Okay. See here, you can see the plasmid is cut open, isn't it? Can you see the cut over here? Here you can see the cut in the bacterial plasmid, and then we have the gene of interest which we have taken from the whatever gene we want. It might be the gene for insulin or for any human growth hormone or any other gene which we want to express, right? So for the gene, there will be several genes for several characters, and we can utilize this recombinant DNA technology for various needs. Okay, so this is the gene of interest which we have extracted from the whichever source we have. That gene is put into this bacterial plasmid. You can see here, first the bacterial plasmid is extracted from the bacterial cell, then it is cut over here. After cutting it, we are putting this gene of interest over here, and then you get the recombinant DNA molecule over here. So this is the recombinant DNA molecule which has the plasmid DNA and the gene of interest. Got it? Then you can see this recombinant DNA molecule which consists of the plasmid and the gene of interest together combined, that is the plasmid molecule or the vector molecule along with the gene of interest, both of them combined together is called as the recombinant DNA molecule. That is inserted or put into the bacterial cell. Okay, so this is the bacterial cell. Now this recombinant molecule is inserted into the bacterial cell. Okay, so you can see after it is inserted, as and how the bacteria divides, even the plasmid also, that is a recombinant molecule which is inserted inside the bacteria, that also starts multiplying, producing several copies of itself. So you'll have a huge colony of bacterial cells from which will express that gene within itself. So that gene which we have inserted over here, that will get expressed and it will start producing the hormone or the protein which we want. Then from those bacterial cells we can extract that protein, we can process it further in pharmaceutical industries and we can utilize it for the whatever needs we have. Got it? I'll just show this full picture once again. So here you can see the extraction of plasmid. Here you can see how the DNA which we need, it might be from, it need not be that the gene of interest is extracted from human beings alone. You can extract it from some plants. Say for example, you want some drought resistant plants or some other plants with specific needs. Okay, so that gene from some plant which it is having, that you can extract from that plant and you can integrate it into another, integrate it over here and produce that whatever we need. Okay, I'll be explaining the applications so that you will understand it better. Fine. So you don't think that recombinant DNA technology has applications only in human beings. No, we have applications of recombinant DNA technology in human beings and in animals, in plants. There are various applications which I will discuss slowly. So here the gene which we are extracting need not be from human source alone. It can be from plants, it can be from other animals, and it can also be from other bacterium or other microorganisms also. Okay, so we can extract the gene of interest from some other microorganism also. That is also a possibility. So this need not be an animal source or a human being source alone. Got it? So here you can see that we are extracting the gene which we need, and that gene we are cutting it, or whatever particular, okay, complete DNA we are extracting over here, and from that DNA we are cutting off the tiny portion which we are interested in, and that only we will combine it with the plasmid molecule and to produce a recombinant DNA molecule. This recombinant DNA molecule is inserted into the bacterium, where the bacteria also divides and also the plasmid also starts replicating and producing multiple copies of itself within the bacterial cell, thereby expressing the gene which we have inserted to produce the protein which is of our interest. So we can extract the protein from the huge bacterial colonies and we can process it further and utilize it for our needs. So, did you all understand how recombinant DNA technology this is PCR, I'll be telling you at a later point of time. Okay, so as of now, in very simple terms, did you understand what is recombinant DNA technology? So recombinant DNA technology has various applications. Okay, so using genetic engineering or recombinant DNA technology, we can produce various chemicals, polymers, amino acids and various food additives to develop livestock and other animals. Okay, so here I have given you in your material, I have given you simple, I'll just show you 1 minute. So here I have shown you the picture for human growth hormone. So first I explained you with the example of how insulin can be commercially manufactured without relying on animal sources for insulin. So earlier we relied on slaughtered animals and cadavers to get insulin from them and administer to the diabetes patients, but later using recombinant DNA technology we could make commercial insulin using recombinant DNA technology. So first I explained you with the example of insulin, right? So next is human growth hormone. So here you can see this is the gene for the human growth hormone, okay, that is inserted into this plasmid and further it is inserted into the bacterial cell E. coli. So what is human growth hormone? Many a times you will see these advertisements of Complan, isn't it? So children will be very short. So their parents or friends or somebody will advise them to go drink lots of Complan so that they will grow very fast and overtake their peers. Isn't it? So what happens is human growth hormone is a hormone which is necessary for our proper growth and development. But when the hormone is less in quantity, then the children do not attain the normal growth and development according to their age. So in those cases, earlier they used to treat them with hormones from animal sources. Again, just like insulin, they used to take growth hormones from other animals, slaughter animals, and they used to administer it to the people who were suffering from dwarfism. So however, it was not as effective. Further, even in this case also what happened, there used to be a lot of allergic reactions. So then they started synthesizing the human growth hormone using recombinant DNA technology. Okay. So here you can see I already showed you. Here see this is the gene for human growth hormone, this gene, and it is taken and it is inserted into a plasmid and that plasmid is inserted into a bacterial cell. Okay. And then finally that expresses and we get the human growth hormone. Fine, like this you can find various examples for applications of this is recombinant insulin. So actually speaking, insulin is not a single it is made up of two polypeptides. So what we do is, just for your example for explaining, earlier earlier for explanation purpose I showed a single gene as a gene of interest for insulin. Actually speaking, insulin is composed of two polypeptide chains like this which are linked together. So this complete two polypeptide chain together forms the insulin hormone. So what we will do in laboratory is, that is, we will isolate the gene which codes for this polypeptide and we will produce that, and again we will isolate the gene which is responsible for this polypeptide. We will produce that also using recombinant DNA technology, and then once we get these two, we will join them in the laboratory conditions and we produce the fully functional insulin hormone. Okay. So this is what you can see. See, we have two chains in insulin, A chain and B chain. Okay, insulin A chain we produce separately. See, we take out the gene which is responsible for producing A chain. We take the gene which is responsible for producing B chain of the insulin and we put them into the bacterial cell using plasmids, which here you can see first, which see, this is the gene which codes for the A chain of insulin. This is the gene which codes for B chain of insulin. So we take that, okay, we put it into a plasmid. That plasmid is inserted into different bacteria. Then see, here you get the A chain of insulin, here you are getting the B chain of insulin after doing recombinant DNA technology experiments. And then you see, here you have the A chain, here you have the B chain. We are combining both of them over here. See, both of them we are combining together to form the functional insulin. Got it? Then Similarly, we can make recombinant vaccines also. So this is a vector. Okay. Again, the mechanism is the same. The basic line is the same. You take a vector molecule, you take a gene of interest, you combine both of them to form a recombinant DNA molecule. That recombinant DNA molecule is inserted into a bacterial cell or a host cell where it replicates and it produces multiple copies of itself. At the same time, the bacteria also multiplies, producing several colonies of the bacteria where the gene which we inserted gets expressed. From that, we can extract the protein and we can utilize it. So, be it for insulin, for human growth hormone, for monoclonal antibodies, or for any other, whatever needs we have, be it for chemicals or polymers, for amino acids, all of this, the process is the same. Okay, this is how recombinant vaccines are produced. Fine. So we have various applications of recombinant DNA technology. It is used to produce human growth hormone, to produce recombinant vaccines. Okay, we have also produced recombinant vaccines for hepatitis B. Okay, hepatitis B virus using recombinant DNA technology. And then it is also used for producing monoclonal antibodies. So monoclonal antibodies are effectively used to control infectious agents and in tumor cells, and to control the multiplication of tumor cells, monoclonal antibodies are very much used. So we can produce monoclonal antibodies using this recombinant DNA technology. Okay, so monoclonal antibodies are used for diagnosis of infection, used in tumor therapy, that is for in cancer therapy, and then it is also used for imaging the tumors for radiotherapy. So these are the various applications of monoclonal antibodies. Okay, then we can also use recombinant DNA technology in plants. Okay, we use recombinant DNA technology to develop livestock and other animals that have enhanced genetically determined attributes. Say for example, some cow wherein it has very good, it has very good features, but it is not capable of producing more amount of milk. So we can take that gene from some other animal, okay, and we can insert it over here and we can do that. Okay, so not exactly that. I gave you a layman's example. Don't write those examples. Just to help you understand that you can develop the livestock and other animals that have enhanced genetically determined attributes. Say for example, you have a plant which has very good attributes, whereas it lacks in some feature. That feature alone you can take from another source. You can insert it into this plant and you can make it give much better. Be it much better, it can be pest tolerant, or it can be drought tolerant, stress tolerant, or it can produce much better crops, something like that. Okay, so to facilitate removal of pollutants, waste materials. Okay, then microbial genes are transferred to plant system to obtain disease resistant plants. That is what we did in our BT cotton, isn't it? It is resistant to the bollworm. What is BT cotton? What does BT stand for in BT cotton? BT cotton. We've heard so much about BT cotton and why it is not good. There have been so many discussions, arguments, everything. So let me know if you want me to make a lecture on few important topics in biotechnology, I will be able to do that for you. Okay, because now if we start discussing about BT cotton and a few other topics in biotechnology, that itself will go for 2-3 lectures. So we cannot discuss that as part of this lecture. However, it's not much needed for your anthropology also. However, in GS for science and technology, you must have definitely studied a few important topics in biotechnology, right? So just let me know if possible. I will make one lecture for you on important topics in biotechnology for your GS. Okay, so here in BT cotton, BT stands for Bacillus thuringiensis. Bacillus thuringiensis. Fine. So, we use recombinant DNA technology or genetic engineering to produce disease-resistant plants, insect-resistant plants, okay, to increase the shelf life of fruits and flowers. What is the meaning of shelf life? Say you buy tomatoes from the market. So already it would have taken 1 or 2 days for the tomato to reach the market. So the farmer would have harvested the tomatoes, then he would have transported it from his village to the nearby town, and from that town it comes to your city. So it takes probably 1 day or 2 days to reach the market. After you buy it from the market, you keep it in your kitchen or in your shelf. So how long it is going to be fresh? Maximum it will stay fresh depending on whether it is very ripe or it is still not so much ripe. It will stay for 3 days, 4 days, or maximum 1 week. After that it will start rotting, isn't it? So this is called as the shelf life of a vegetable or fruit. So using recombinant DNA technology, you can produce fruits and vegetables which have increased shelf life. Okay. So again, there is what goes into the ripening of this. What happens? What exactly happens when fruit or a vegetable is going through the process of ripening? Why some fruits ripen early? All this also we can discuss later if you want. Then to increase the shelf life of cut flowers. You know about cut flowers, right? All this which we use for decoration, roses. Carnations, zerbera. Lot of these cut flowers are utilized in a decorative. When you go to restaurants, you can see them using these flowers for decoration, and even when you go for functions like marriages, there also you can find elaborate decorations, right? So these are the cut flowers. So again, there is a shelf life for cut flowers also, right? So it is said that Bangalore is the highest exporter of cut flowers in Asia. Again, I'm not 100% sure, but at least in India, I'm sure Bangalore is the highest exporter of cut flowers in India. Then we can also utilize genetic engineering to produce edible vaccines, monoclonal antibodies, flavoring agents in case of plants. Then in terms of animals, you can improve the livestock by increasing the growth rate, milk yield, all that. And then using microorganisms in genetic engineering, you can utilize it for production of proteins, insulin, vaccines, interferons, human growth hormones, and so on. Okay. Which is the first commercially produced recombinant insulin, and the right answer is humulin. It was developed by Gentech, a company called as Gentech, and it was licensed and marketed by a company called as Eli Lilly. So the company name was Eli Lilly, which marketed this product, and the insulin name was Humulin. Okay, it was done in the year 1982. So long before we were all born itself, we had the first commercially produced insulin hormone. Prajakta gives the right answer. Then Kalpana Nayak has few questions. How the gene of interest is exactly extracted, and how we know which gene will produce A or B chain of insulin and all. So actually, if you have to go to that depth, I will suggest you few genetic engineering textbooks which I referred during my college days. You'll get an in-depth understanding of that. Otherwise, if we start explaining that now, it will go for how the gene of interest is extracted. That itself goes to several lectures. There are various methods by which it is done. Okay, so don't worry, Kalpana, for your doubts, I will share few articles on the WhatsApp group. Fine, that will, because I cannot send my textbooks to you. Okay, I will search online, which gives in-depth knowledge about the various methods of extraction of gene of interest, and how exactly we can identify this A or B chain of insulin. All these things I will share you relevant articles on the WhatsApp group. You can go through that. Okay, further if you have any doubts on that, we can discuss. Fine. I also wanted to tell you about polymerase chain reaction. Before we move into the other topics, let me give you a brief understanding of what is polymerase chain reaction. I'm sure Sir would have also discussed that in another topic. However, I'll also explain a little bit about polymerase chain reaction. I think you have the materials for that already. So what is PCR? PCR is called as polymerase chain reaction. Polymerase chain reaction. So polymerase chain reaction, or PCR, is a technique that allows the amplification of a particular DNA sequence and is able to generate innumerable copies of a specific DNA sequence. And this polymerase chain reaction was designed and developed by Karamolis in 1980. Karamolis in 1980 designed and developed this. So what is polymerase chain reaction? See, suppose in some cases we have very tiny amount of sample of DNA, and if we want to conduct many tests with that, we need multiple copies of that, isn't it? So that time we can use this polymerase chain reaction to produce several copies of the sample DNA which we have. Say for example, you take the case of a crime scene where the person who conducted the crime, he has by mistake, by chance left some either his hair follicles or his skin tissue or his nail samples, or some, say in case of rape scenes, you can find the semen, right? So like this, some criminal activity has happened and he has left behind some evidence, like evidence in the sense here some sample, some body sample, be it hairs or nails, nail clippings, or even the skin tissue. In most of the rape cases and all, it is said that the woman would have tried so hard to get rid of him and try, she would have clawed his body. So mostly they will extract the skin cells from the fingernails. She would have clawed onto his body. So in that process, she would have managed to get some skin cells inside her nails. So usually that skin, the skin cells from the nails are taken, and from that DNA is extracted. So in all these cases, and not just in cases of crime, in many other cases also, when we have very tiny amount of sample of the DNA, but we have to conduct tests. We cannot utilize that tiny sample and we cannot conduct several tests, right? So in these cases, what we can do is we can conduct this polymerase chain reaction. So if there is one copy of the DNA, okay, one copy or one tiny sample of the DNA, we can make multiple copies of that using polymerase chain reaction. And using this multiple copies, we can do several other tests, isn't it? So polymerase chain reaction is a technique that allows amplification of a particular DNA to produce several copies of that. So like what we do with our materials, right? Like you are start preparing for the exam, you have some material and you want to share with all your friends. So what you will do, you will take that single material and you make several copies of that, so that you can share it to all your friends, be it 10 members or 20 members or 100 members, whatever it is. So you are making multiple copies of that. Similarly, using polymerase chain reaction, we can amplify a particular DNA sequence and we can generate innumerable copies of specific DNA sequence. Okay, so this polymerase chain reaction has three important steps. First one is denaturation. The first step of polymerase chain reaction is denaturation. So what happens during denaturation is we increase the temperature. Okay, so we increase the temperature to 96 degrees centigrade so that the two strands of DNA will uncoil. So we know this is the structure of DNA, right? There is two coils which are two strands which are coiled like this. So when we increase the temperature to 96 degrees, they will uncoil and become separate strands. So this is the first step of denaturation. The second step is renaturation. Second step is renaturation, or it is also called as annealing. So what we do during this step is, after the two strands have separated, now we reduce the temperature to 55 degrees Celsius. Okay, so polymerase chain reaction, the first step is